So in today's lesson, we're going to begin to delve into the nitty-gritty details of photosynthesis. But before we do that, I want to give you a brief overview of the process. So basically, what is the overall intent of the process and what are the stages that are involved? So before we do that, I just want to remind you of a couple of relatable ideas. So let's just first talk about cellular respiration. So the goal of cellular respiration was, of course, for us to start with glucose, which is a large molecule, to break that down, and we ended up with CO2, and we also ended up with, some other products, but, we took that large glucose molecule and broke it down. And in addition to that, we were able to produce a lot of ATP. So cellular respiration we could characterize as an exothermic reaction. Now, photosynthesis is sort of an inverse process to cellular respiration. So we should expect a lot of these ideas to be familiar, but perhaps they're happening in sort of a reverse process. And that's not untrue, right? In photosynthesis, what we're going to do is we're going to be taking CO2 and we're going to be somehow combining them to make glucose molecules. Now, forcing, this goes against sort of the idea of how nature works, right? We are taking small pieces and organizing them into larger pieces. So that is going to require some ATP. So we're going to need somehow some ATP to come in and make that happen. Now, so this we could characterize as an endothermic process. But the reality is that ATP is going to have to get made somehow. And how it happens in photosynthesis is by capturing light's energy to make ATP. And that pretty much sums up how photosynthesis happens. There are two distinct stages, the first of which is called the light reaction. And the goal of the light reaction is to take light energy and store it in the form of ATP. Now, the second phase of photosynthesis is sometimes referred to as the light-independent reaction, or as the Calvin cycle. It's a cycle where we take our CO2 and join some CO2s together to make what eventually becomes sugar. But as I mentioned, that is taking disorganized pieces and organizing them, which is going to require energy, hence the endothermic process. And how we're going to do that is we're going to take the ATP produced in the light reaction in order to join our CO2 molecules to make sugar eventually. So that is basically the process by which photosynthesis takes CO2 and produces sugar. Those are the big ideas. And so in the next few lessons, we're going to be taking those ideas and breaking them down further. So let's write a little bit down about that. So again, we're going to break down photosynthesis into two main stages, the first of which is called the light reactions. And of course, it's called that because this is the stage that depends on light availability, because we're going to be capturing the light energy and storing it in the form of ATP. So where is this all happening? It's happening in the thylakoid membrane. So you'll remember, just to quickly draw here, we'll have our double-membraned chloroplast, and then we'll have our thylakoid discs arranged in grana. And so really, the light reaction is happening right in that part of our chloroplast. There we go. Nice. So the light is going to be captured by the photosynthetic pigments that are in that thylakoid membrane. So you'll remember that, of course, embedded in that phospholipid bilayer that is the membrane of our thylakoid disc, that's where we're going to see our chlorophyll molecules. And I'm not going to do a great job of drawing this, but you'll remember that right in there we have our porphyrin ring and our phytyl tail that's embedded there. So that's where we're going to find the photosynthetic pigments that are going to allow for our sunlight to be captured and then utilized to produce ATP. So we're going to get into more detail about the mechanism by which that happens, but let's leave it for today by saying that we're going to see some clusters of those pigments in that thylakoid membrane. They are referred to as photosystems. We'll have a whole separate note on what a photosystem is and what it's comprised of. But just for now, know that it's located in that thylakoid membrane and that it's utilized again. We're going to take the light energy and convert it into ATP. And we're going to see some molecules that look slightly familiar along our joyous ride that is photosynthesis. We have NADP plus becoming NADPH. We're reducing that molecule. So again, that is just another electron carrier that is specific to photosynthesis, but not unlike what we see in cellular respiration. So again, we're seeing another element of photosynthesis that should remind us of cellular respiration. We have a cycle, a cyclical process. So of course the Krebs cycle is what was cyclical in cellular respiration, but in photosynthesis it's referred to as the Calvin cycle. It's where we take carbon and we fix it into glucose. So again, if we're taking a bunch of, you'll remember that we have, generally speaking, in photosynthesis, we have to take 6 carbon dioxide molecules and somehow get them to form one glucose molecule, right? And so we're going to use the ATP that was produced in our light reactions in order to fix these carbon dioxides into glucose. Okay, and so what can we say about that? Well, we can say that first of all, it's occurring in the stroma. So if we go to, we think about our chloroplast again, we're not dealing with our grana anymore. We're just dealing with this sort of space in between our grana in our chloroplast. So as was the Krebs cycle, the Calvin cycle is just cyclic in nature, and we're going to see enzymes catalyzing each stage in that cycle. And what is powering the cycle? Well, it's the ATP we produced in the light reaction and the reducing power of our NADPH that's going to help us make that glucose. So as we did when we discussed our overview of cellular respiration, I want us to give sort of the big picture, big ideas, and the source of destination of each molecule and atom within the process. So we know the overall chemical equation for photosynthesis shows carbon dioxide and water plus some light energy with chlorophyll helping us to do that. We're going to be able to make some glucose and some oxygen. Now what we see here is, just to make sure you understand, usually the equation for photosynthesis cancels out the water that are used with those that are produced, and we usually typically only see 6 CO2 or 6 H2Os in our reactant side and no H2Os in our product side. But if we're going to show what's actually happening, we put in 12 waters and end up with 6 out, so there's a net use of 6 waters. Okay, so I've deliberately colored out the atoms based on where they end up in the process, just as an FYI, and we're going to talk more about water and oxygen's relationship. So note that when we're talking about CO2, it should make sense that all of the carbons from our 6CO2 should end up in glucose. That seems a bit like a no-brainer. But one thing that's worth noting here is that the oxygen that is produced, here the O2, is not coming from CO2 at all. It's strictly coming from our water molecule, right? So the O2 that's produced, those oxygen atoms came only from the water that was used as the reactant, not from our CO2. Now, how did scientists figure that out? Well, let me explain. So it used to be, in fact, that scientists believed that the CO2 was once the source of the O2, and that wouldn't be way out of line. You can sort of see how that might be an assumption. But what scientists did is they made use of an isotope of oxygen, and they used O18. So you'll remember that oxygen, the most common isotope of oxygen is O16, but here we're looking at a heavier version of oxygen, and that allowed scientists to be able to trace it through the metabolic process that is photosynthesis. And so in using that radioactive tracer, they were able to show, well, where did the O in water, where did that O18 end up? So what they did was they tagged the water in a photosynthetic process with that O18, and they saw where it ended up. And so it ended up entirely in the O2. And so they found that if they did the same thing with CO2 and tagging it, then the oxygen didn't end up with any O18 in it at all. So that was able to prove to them that the oxygen, that the water was the sole contributor to the final oxygen product in photosynthesis. All right, which is kind of cool. Cool way to run an experiment. So, by the way, I should mention to you that H218O in this case is referred to as heavy water, although there are different ways that we can just make water to be considered heavy. Other ways by changing the hydrogen in water, but in this case, heavy water refers to the oxygen being the heavier isotope that's possible for water. So, that pretty much sums up our overview of photosynthesis. Tomorrow we're going to talk about photosynthetic pigments and how they absorb light, and then we're going to break down the light reaction and the light-independent reactions of photosynthesis.